few months back, my mom fell ill with pneumonia. She had to go to the doctor to get a medication, and they gave her a puffer. So she took this puffer for the week, and after the week, she realized that her condition started to worsen instead of get better. So she went back to the doctor, and they gave her another medication, and the same cycle repeated for the second medication and the third medication, until finally, on the fourth medication, did her condition actually start to get better. So this got me wondering, why couldn't the doctors have just prescribed her the right medication in the first place? And so I researched deeper, and I realized that everybody responds differently to medication. So while you might have a normal response to a drug, I might have a toxic response, and somebody else might have no response to a drug. So this blew my mind, as I realized that what we're doing with medication today is just plain out wrong, and that we should be personalizing it to everybody's genetic code. So why do you respond differently to medication than I do? Well, that's because of certain genetic variants in our DNA. So for example, a small a can change not only why you might have freckles and why I might not, but also why a drug might have optimal effectiveness on you and not on me. So these specific mutations code for enzymes and proteins. In pharmacogenomics, we look at two main enzymes. First, the VKORC1s, which are vitamin K expedites and help with the metabolizing of blood clot medications for optimal effectiveness. And secondly, we have the cytochrome P450s, which are the most prevalent drug metabolizing enzyme, and about 80 to 90% of specialized medications out there today that are actually effective have this enzyme. So to put this into perspective, let's say somebody's taking codeine for pain relief. So the active form of that is morphine. And just keep in mind that codeine has a higher toxicity than morphine. So moving on, person A has allele 1 from father and allele 1 from mother, therefore coding for the CYP2D6 gene, allowing them to have an extensive metabolizer, meaning that they would need to take a medication with high toxicity or high dosage. So therefore, they would take codeine over morphine. Whereas in person B, they have allele 4 from father, therefore allowing them to be an intermediate metabolizer. So therefore, they have to take a medication with lower dosage or lower toxicity. So therefore, they would take morphine over codeine. So with all the knowledge I gathered in pharmacogenomics, I decided to start my own experiment, in which I tested for which blood clot medications had optimal effectiveness on which genotypes. So in my experiment, I took 3 main blood clot medications: aspirin, warfarin, and clopidogrel. And I chose these because they all had the same purpose, which was to alleviate platelet in the veins, thereby allowing red blood cells to pass through. So I split my process up into 3 steps. First, I put certain genes in an Illumina simulator. Then I figured out the variant associated with my genotype and medication. And lastly, I figured out how the drug was metabolized throughout the body, and in the end, if it was effective or not effective. So first, putting certain genes in an Illumina simulator. What we can see here is the specific genes in my Illumina simulator along with a bar on the top right-hand corner. So what that shows is how much I've covered about the medication and genotype that I needed. But what I specifically want to point out here is the table at the bottom, and how that what we're looking at here is the VKORC1 enzyme. So now, we'll be figuring out the variant associated with our blood clot medication and our genotype. So what we see here is a genome browser. Now, this has all the information on our specific enzyme. And while this might look complicated, that's because it is complicated. And so what I specifically want to point out is that we're looking at the left side of the chromosome, indicated by that red vertical line. And as we look at different variants, that red vertical line will change to the right-hand side. And secondly, the variant number. So this variant is actually very hard to find, as it has to be specialized to our specific genotype. So in our case, genotype GG, the medication that we're testing for, so in our case, clopidogrel. And lastly, the ethnicity has to stay the same, as if you might have a different ethnicity than somebody else, that could play a factor in how someone might respond to a drug. So in our case, the ethnicity is Japanese. So now we're going to figure out how the drug is metabolized throughout the body. So what we're looking at here is all the information on the variant associated with our gene. Now what I specifically want to point out is how we're looking at the ABCB1 gene. Now those bars, they represent the metabolic process and each of the steps. So we'll only be looking at the first 5 bars. But what the first 2 orange bars mean is that when our drug is first inhibited in the body, it is calling on certain channel proteins and certain receptors to therefore break the blood barrier and allow the platelet to decay. Now we can tell that our channel protein is being effective by the large green bar, as it is very high compared to the others. Now, the last 2 bars are when the calcium is also being activated, therefore decaying at the platet, and this is the result of the channel proteins and the receptors. So what I figured out in the end was that genotype GG responds best with clopidogrel. So I just specialized medication to a specific genetic code and genotype. And that genotype AA, AG, and AA and AG respond better with aspirin and warfarin. So this can not only be done for blood clot medications, but also for pneumonia drugs, cancer drugs, and even deep vein thrombosis drugs. So the question here is that if the technology is there, then why aren't we doing it? If a 13-year-old boy can specialize a specific genotype to which blood clot medication will have optimal effectiveness, then I'm sure that we'll be able to disrupt the pharmaceutical industry by doing this for every single drug out there. Thank you.